I was just told that this meeting's being recorded, so. No? Oh, everybody's acting normal now. Funny that. All right, go leave me in peace for a little bit. Okay, so lab begins. Sorry for being a little bit late. Had to deal with this lot and puppies and stuff. Okay. Pepper, come here. Sorry, I just gotta wait until this lot leave. Yeah, she's very cute. Leave. Go on. Whoosh. All right. Okay. Distractions over. So before we get started on what we're going to do today, anyone have any questions about the assignment and stuff due tonight, which should be the one on PCR? There we go. Up. Come. There you go. Some obligatory puppy shots for people. There's the older one. And down there, that's hard to, is the little one. Being super cute and snoring. It's proud, as idyllic as you can get for teaching, as far as I'm concerned. Other than it being like some rustic log cottage in the mountains. Yeah, she's funny. She was our first. Oh, yeah, good job there, dog. Go lick your bits while people are admiring you. Okay, so anybody got any questions about last Thursday's last labs assignment? Right. Also, there's time for any questions about uh, anything that might be on exam 3, which actually should be open by now. So you can't ask me specific questions if you're taking the exam right now, because that'd be a bit cheeky. But general stuff. I'm stroking the dog while I'm listening, just in case you wondered while I was leaning off the screen. Speak now or forever hold your peace, as they say. I'm getting stared at by my brown dog because I didn't take him out for a walk this morning. That's a reproachful look. Why didn't you take me for a walk? I was doing something else. Trying to teach a little bit how to walk, like on a leash. That's exhausting. It's like a workout for me and for her. Okay, well, if anything pops up, then just ask. I don't mind being interrupted. So last lab, we worked on the principles of PCR. And so if you look at the lab for today, the folder, you'll see a gel image. And so essentially what you do after that is, one hopes, you know, if the magic fairies did their bit, or actually in one lab I worked in, they had a rubber chicken that was suspended over the PCR machine for good luck. If all that works, you should get tons of DNA of the size that you want, in our case 401 base pairs. What else are you going to have in that little tube in addition to your target DNA, the thing that you're trying to amplify, or your PCR product, the thing that you have amplified? What else are you going to have in that mix? Yep, good. You're going to have lots of salts, magnesium specifically, but also probably others as well. I know magnesium is there because that's an important one for the PCR to work. What else are you going to have? Think of the different components that go into a PCR that we talked about on Thursday. They're all going to be there still. So you're going to have salts from the buffers. What else are you going to have? What other things do you need for PCR? Obviously, you're going to have DNA. What else? Electricity? That'd be pretty cool if you did. That would be provided when you do the gel electrophoresis to see if this works. But in the actual PCR tube, one would not expect to have electricity. What makes the PCR product? What makes the DNA that you're hoping to get your product? What's that enzyme called? It's a squishing of the name of the bacteria in which it was found. Yes, a DNA polymerase. Anybody remember its name? First letter comes from the genus, which is thermus, as in hot. Where was that bacterium isolated from? No, actually, Dipper. It's a good idea, but that should all be gone. All the cell material. Yes, Maria, TAC, Thermus aquaticus, DNA polymerase. To answer Dipper's question, all the cell material should be gone from the DNA as a result of the DNA purification process. So you should only be putting DNA into your PCR mix. All right, so we've got TAC, DNA polymerase, we've got salts, buffer. What else is really important for your PCR to work? What tells the DNA polymerase that that's the bit of DNA you want to amplify? It's also something that's made, although an RNA one, during DNA replication to start the whole process. What are those called? Also found in bullets to kickstart the whole process. Same kind of idea. Yeah, that's the one, Maria. Primers. Yeah, you beat Robert. Too slow. You snooze, you lose. Yeah, primers, right? So the primers are the bit that kick off the DNA replication, but they're also the bit that specify exactly which bit of DNA is being amplified. So let's imagine you take that PCR, right? You're going to have gangbusters amount of DNA in there that you want, but you're also going to have buffers, salts, DNA polymerase, and primers. Now, we want to sequence that target DNA because that's the whole point of this, right? We want to sequence it, find out what changed between our normal brown flies and these yellow flies, right? What's the sequence change responsible for that phenotype? So do we want all that extra stuff in there when we send it off for sequencing? Do we want primers and buffers and enzymes and stuff? No, you don't, because they're going to interfere with the downstream process. So before we even think about, yay, time to go send off stuff for sequencing, we need to do two things. One, we need to clean up our DNA or PCR product by getting rid of buffers, primers, and tack. So that's the first thing that we need to do. We also need to make sure it works, right? Right, which isn't a given, right? I mean, this particular PCR works very reliably, right? It's pretty rare that it doesn't work. But, when you do this for yourself later on, you may find out that sometimes it just doesn't work right. So, did the PCR work? And so we have, first, what we're going to do is what's called a PCR cleanup. And that basically uses the same principles as our DNA purification. Essentially, minus all the other stuff, minus all the stuff that is to do with, cell debris, protein contamination, stuff like that. Right, and so that we would be using essentially the same deal. We'll be using a spin column, right, which we used for DNA purification. But we don't have to do a homogenization step, right, because we're not dealing with tissues. We don't have to do a lysis step because we're not dealing with cells. And we don't really have to do a proteinase step because our DNA should not be complexed with protein, right? It's not going to be chromatin. It's just going to be a PCR product. So all we need to do is to put our DNA and bind it to the column. Let's do that with letters instead. And exactly the same principle as before, you pipette it out of your PCR tube and mix it with some chemical that helps it bind, and then you spin it through a column, right? The DNA will stick to the silica. And you wash all the other stuff off. Right, that's the primers, the buffer, the salts, the protein. So one thing you might be thinking as well, the primers are made of DNA, right? So why don't those bind to the silica column, right? And the answer is that those bits of DNA are too small. They don't actually bind very well, so they're very easily washed off. And then once you've got everything nice and clean and dry, you elute the DNA. It's kind of similar as to how we did it before with the DNA purification, using ultra pure water or buffer. We typically use ultra pure water because we don't want any salts or anything like that in our DNA for sequencing. And then the second one is basically run it on a gel. Right, and that's how we visualize what we have or not. So if you look here, Go on, off you disappear. Nope, nope, nope. Ooh, hang on a second. Let me go back to where it was I wanted to be. Not that one, that one. So essentially and this was a slightly older one which used a different gel imaging system, which is why it appears colored rather than black and white. But when you do that, this is what you get, right? So this is actually before purification. And the reason that I know that is if you kind of look at it kind of funny, you know, particular angle. Yeah, I know. What you'll see is a very faint band down here. You can see it a little bit more clearly in this one too. Those are the primers, right? So that's the leftover primers that weren't used to make DNA. And this really bright band is your PCR product. And so this worked fabulously. Remember too, to how the DNA purification looked. Let's see if I can dig that out. Bah, it's not on here. It's in here. Well, it's supposed to be. Oh, here we go. I'm sorry, kind of looking at the wrong folder. So this is the same marker, about the same amount of DNA. Let's get this one. Be easier to do it this way. And compare that to that. So if you look at this is our genomic DNA. So this is the DNA from what, 3-4 flies, something like that. And it's fairly pale, right? It's fairly weak kind of signal, right? It's there, but there's not a huge amount of it. And you compare that to this, right? Same amount of marker. Essentially, you're looking at kind of equivalent brightness to this band down here, or actually a bit more. And now you can see the difference that PCR makes. You have a huge amount of DNA now, and that DNA is just the bit that you want, nothing else, right? There's very little genomic DNA there. It's almost entirely what you're interested in. So just go and look at that again. You can see the difference. So we're going to clean that DNA up. We're going to basically elute it in some nice ultra pure water, which isn't going to interfere with any subsequent step. And then we're sending it off for sequencing. Now I've got a nice little PowerPoint prepared for you, which you can also look at your leisure, if you're American. And so we're going to use something called Sanger sequencing. And Sanger sequencing figures out every single base, more or less, apart from the ones at the beginning, in your PCR product. And it does so in a really cool way. I'll actually show you what it looks like. So this is what a sequence read looks like, right? Series of bases down here. And this is what's called an electropherogram, which is a really cool word. And you can kind of zoom in. And this is quite literally a readout of the brightness of each individual base as it comes out of the sequencer. Right, so this is a plot of brightness, which is kind of how tall the peak is, and color. And the color is what determines what base is present at that point. And so you look through it, Gets kind of nice, a lot cleaner as you get into it. Each of these individual peaks corresponds to a base. And again, this is called an electropherogram. Oops. Let's get back to the chat. Ah, dang it. Where be the chat? And so it's not actually necessary in terms of analyzing the sequence, because what you care about is, this thing down here. This is actually in Eugene. This is the software you'll be using next week. But this is the actual readout that is made which is used to determine what that sequence is, which I think is pretty cool. Kind of science nerd type stuff. Okay. So let's get back to sequencing. So the idea here is that we want to figure out what bases we have in what order for our PCR product. And our PCR product is, we hope, amplified from either yellow or brown bodied flies. So somewhere within that sequence should be a change in our yellow flies that result in those flies being yellow. How do we figure out what the sequence is? And so this is what we use, something called Sanger sequencing, after the person that kind of developed this technique. And Sanger sequencing is what was used to sequence the human genome and the worm genome and other genomes initially during like the early days of genomics, which was actually before some of you were born, which is kind of crazy. I was around that time. Makes me feel a little bit old. So back in, the end of the 90s and early 2000s, this was how sequencing was done. It's not actually done that way for large amounts of sequencing anymore. We use something called next generation sequencing, which does huge amount of sequence data in a single run. But we still use Sanger sequencing for sequencing individual samples. It's a lot more cost effective. And so basically the principle behind Sanger sequencing is something called, well, two actually. Sanger sequencing. Something called dideoxy chain termination, which we'll get to in a second. And two, fluorescent labels. Yeah, I think I spelled that right. So we need those two things. One, we need to stop the DNA replication at a given point. And two, we need to be able to determine whether that stopped at an A, a B, a C, or a G, right? So these individual colors are actually fluorophores, right? So that's the actual color of the light, which is produced by that bit of DNA. And so the dideoxy sequencing, you'll remember deoxyribonucleic acid, or DNA, is called such because it has a 3' OH, right? You have to have this 3' OH for adding another base to that, right? No 3' OH, no base gets added. And so most of the nucleotides in each sample that you're doing your Sanger sequence with essentially you're doing PCR, right? But instead of just putting all normal nucleotides in there because you want loads of DNA, for each tube you're going to put in a little bit of, oops, sorry, I got to move you out of the way, Robert. Stick up there. You're going to put in a little bit of one of the bases, an A, a C, or a G, or a T, which does not have that OH. Right, so I don't know the percentages, but let's say 1% of the A's in that particular reaction have no OH on them. So every time you come across an A for your PCR, there's a 1 in 100 probability you'll incorporate one of these kind of dud nucleotides. And that will result in replication stopping at that point. But it might not, because it might incorporate a normal one. And so what happens when you do this is every place you get an A in the sequence, you'll get a different size fragment. Because for some of those reactions, that dideoxynucleotide, that chain terminator, right, the one that stops everything, was incorporated. Other times it wasn't. So in this one, shortest one, it got incorporated early and stopped it. For other ones, it got incorporated later and stopped it once it had already got beyond a certain length. And so essentially, if that happens every time there's an A, you'll have in your tube a collection of fragments that all end in an A. So you have a really short fragment, say down here, right? That's 2 bases long, ends in an A. You get a much longer fragment up here. So the A here is normal. Now it stops at this A instead. The next time you get an A, some of the pieces will stop at that size, right? So you'll have a tube full of bits of DNA that all end in A's. They're all going to be different lengths, but they're all going to end in an A. Now, if you run those on a gel, right, a particularly high resolution gel, or even better, if you run it down a tube and you flash light as it goes past, you'll get something that looks like this for A. Oops. So that will tell you every place you have an A. Does that make sense? Because the reaction would have stopped by random chance at each of those A's by incorporation of that dideoxyadenine, one that doesn't have an OH on the end of it. So that would be like this lane here. You can see each band or fragment corresponds to an A. Yes. Actually, they use exactly the same spin column. The liquid that you load onto it with the DNA might be a bit different, so the protocol is not exactly the same, but the column itself is identical. And actually, whatever company you buy them from, they'll all use pretty much the same thing, different color plastics or whatever, but the principle is the same. Cool. So, but as you can see, this particular sequence, right, whether we're looking at, this one up here, or we're looking at the, what it looks like kind of over here, consists of more than just A's, right? There's going to be C's, T's, and G's in there as well. So we basically do this again with the other dideoxynucleotides. Right, I might do with dideoxy CTP, cytosine, in which case every time there's a C in the sequence, which you can see up here, we'll get a fragment. As you can see down here. So there are more C's, more fragments. And if we do that for all 4 different bases, A's, C's, T's, and G's, and then we run them either on a gel or in a capillary, each one of these different bases has a different color. Now, if we run these and separate them by size, the smallest fragment will be the shortest one. So now we know that the first base in our sequence is an A. The next one coming off of either the sequencer or on the gel is a C. So it's now C. And then essentially what you can do, this was like the old school way of doing it, you'd literally read this gel. My PhD supervisor actually sequenced a gene this way for his PhD, which I thought was absolutely crazy at the time. I guess technology marches on, right? So you can just use your finger and kind of go up this, and you'll see the sequence is A, C, T, G, C, C, A, G, T, G, G, and you'll just read it up, right? That's how you figure out the sequence. Now, obviously, we do this in an automated way now. So we use a laser, a whole mix of these, and we shine a laser on that tube. And so those fragments are going to, again, separate by size. And as they pass through that laser beam, they're going to flash with a particular color of light. And then there's a little camera that records that flash, and that then plots out those colors on this electropherogram. And then the computer basically just determines what the sequence is from that. So that's how we generate the sequence information. What we'll be doing next week is taking that information and then figuring out what it means. And so we're going to be using this piece of software called Eugene. I'd strongly recommend you download this ahead of time. It's a pretty chunky piece of software, right? Open it up, play around with it a little bit. There should be sequence files for you. Move Robert back over there. To play around with. So if you look in the Sanger sequencing and sequence analysis one, there's a zip file here with good sequences for you to play with, and we'll also get some off the net. So maybe open up one of those or two, have a little play around, and we'll use this to take those sequences, tidy them up, and then compare those to what is a known good sequence. Basically, see what change occurs. And that's what we'll do next Tuesday and Thursday. So essentially what we'd be doing today would be packaging all these things up, and then we send them off in a envelope to a company, give them like $7 a read, and they will sample, and they'll send us back the information. We've already got all that information, and so we'll be using those sequences, right, which are called good yellow sequences in the folder, these ones, and we'll be using those to do the analysis instead, rather than your own sequences that you've generated the DNA for. And so that's pretty much it. Obviously, for current sequencing, we don't use this method anymore, like for genome sequencing and the like. We use something called next generation sequencing, which, let's see if I can pull up some cool pictures. Hopefully it's not just going to be pictures of the actual thing itself. Yeah, a whole bunch of I don't want YouTube videos. Nah, not particularly good pictures. It's not something that's very easy to actually take a picture of, to be honest. Yeah. So in next generation sequencing, what they do instead is, you know, glass capillaries or capillaries, like very, very fine glass rods. They stick them all together like so, and to make a kind of about that sort of size piece of glass, right? And they slice it and they etch it. So each of those really tiny, I can't even show you like with my fingers how small those bits of glass are. Each of those is etched to create like a little bowl, little well, and DNA is put into every single one of those. And so now what they do is when they incorporate a nucleotide, whether it's an A, C, G, or T, it flashes. And so there's a camera above which takes a picture every cycle. Rather than doing the whole PCR and then running along a tube, now you're actually looking at each sample as you do the PCR. And every time one of those flashes, it tells you what nucleotide it's had incorporated. And the camera actually takes a picture and the computer remembers what DNA is in what well, and that builds the sequence together. So it's massively parallel sequencing is another way of describing it. And so essentially those advances are what allow us to sequence a genome for about $1,000, give or take. Does anybody know how much in today's money the human genome sequencing project cost? Roughly? It will astound you when I tell you. How much do you think? $100,000? $1 million? How much do you think it took to do one human genome? Yeah, $1 trillion. It was several billion dollars. So just to do one genome sequence, I don't actually know whose genome was sequenced or if it was just a bunch of DNA all mixed together from different people. But I think it was around $2 billion back in 2000. So kind of current money, we're probably looking in $10 million or thereabouts. So yeah, no Dr. Evil and his small cat kind of deal, but a shit ton of money, right? And that's why it took the US, the UK, whole bunch of other countries kind of piling in to get this done. And now you can just go to the doctors, give a DNA sample, and they'll give you a genome sequence, basically. You know, a few days later for $1,000 or $2,000. It's absolutely astonishing. And the reason being is this. There's the. I think that's. I'd actually call that Mr. Evil. Oh, that's a bit rude. Sorry, I didn't mean to put down your cat. It's a very cute cat, super fluffy.
No, I mean, you're not wrong. So
Yeah, he looks like he would make a good kind of prop for some kind of evil villain.
hmm
Definitely. You'd have to like sit on a chair and like, you know, wearing a suit, stroking him, looking evil.
yeah.
Yes, Mr. Bond. And probably with a Russian accent as well, for good measure. See, anyway, things have really come on leaps and bounds since that initial discovery. And it's actually really cool now because they have um, what's kind of colloquially called personalized medicine. And so actually it still does go on, you know, my experience with my kids and others. But the old school way of medicine is, you go to the doctor and say, oh, I've got this problem. And they'll go, I'll give you some of these drugs and we'll see if they work. And then you go back in a month and you go, no, that didn't do anything. And so the doctor will stroke his or her chin and go, okay, let's try this drug, see if that works. Who knows, worth a shot, right? And, until we find something that works. And that's actually how they used to do cancer therapy, or still do for many patients, right? We think that this particular cancer is caused by this particular problem, maybe, and so we'll give you this drug. And hopefully we won't give you so much this drug that it'll kill you, right? It's got a bit of a balance. And obviously that doesn't work very well, right? And so when it's, about hemorrhoids or something, it's like, oh, well, you can wait a month or two. Yeah, it's kind of based on averages, I think is probably the best way of thinking about it. Most people with this particular ailment respond well to this particular drug at this particular concentration, right? Most people, right? As determined by clinical trials and stuff like that. But obviously, most people are not all people. And so there are people where that either does absolutely nothing, or people where that level of drug can be really harmful. Right? I always joke about how, ibuprofen does nothing for me. Like maybe if I take half of a tub of it, it might knock my headache back a bit. But ibuprofen generally, I can't tell any difference after taking it. Other people are very sensitive to it. So if they take, 3 tablets instead of 2 every 4 hours, they could actually have liver problems. And it's the same in cancer. So now the idea is, well, rather than just guessing, what we're going to do is we're going to sequence your genome and we're going to see what alleles you have for particular markers for drug resistance or drug susceptibility. So it may be that you have an allele liver enzyme, right, which metabolizes that drug really effectively. So if you give a normal dose, then the patient will just inactivate that drug before it can do its job, right? Or maybe that drug needs to be converted from one inactive form to an active form. Well, if you have a enzyme that does a really good job of that, you could potentially overdose or have too much of something because your body's doing too good of a job. And so along with immunotherapy, personalized medicine is actually kind of the future when it comes to medicine, right? You're not going to go into a doctor's and say, actually, you might even be treated for a problem that you didn't know you even had based on different genetic markers for different diseases, simply based on your personal genome sequence. And that's way beyond what 23andMe and the like peddle. It's far, far beyond that. That's kind of looking at association of different markers with different traits and ancestry and like. This is actually drilling down into what form do you have of this particular protein? Is it one that works really well, or is it one that doesn't work at all? Is it something that's not going to be affected by this drug, or is something that is going to be susceptible? It's really pretty impressive. And so maybe in my time, but certainly in yours, it'll become fairly routine when you go to a doctor's for them to just pull up your genome sequence. Type in, Robert has extreme allergies to cats. And then it'll come back, based on your genetic information, you like long-haired, fluffy cats, or something like that. I don't know what. But that will be the future, which is it's kind of cool, kind of scary too. Anyway, and that's all been the result of essentially all the huge amounts of money that went into Sanger sequencing to sequence the human genome, which is kind of like a scientific moonshot type project. But also all of these advances that have come over in the last 20 years in sequencing technology. The cost of sequencing has dropped exponentially over time. Just as the power of computing has increased exponentially over time too. So anyway, that's pretty much it for today. The assignment is kind of really based on two things: the PCR cleanup and the principles of Sanger sequencing. You know, how does it work? What's kind of behind it? So you should be able to find all the information you need, both in the lab handout, the PowerPoint, and what we've talked about today. And that's pretty much it from me. I'm all ears if anyone has any questions or things they want to ask about or the like. But don't take too long because my ears are aching. I actually tried, my wife got for her job like these big headphone, like the big cans. I tried that, but all I could hear was myself talking, like in my ears. So I didn't like that very much. So I have these nasty ass foam jobs instead, which make my ears ache after a certain number of hours. For sure, Gaining, go ahead. Is it this lab or previous lab?
It's this lab.
Mhm.
It asks what size the yellow sequence would be in BP if we were to do this in person. And I think I got it wrong, and I'm kind of confused on that one.
401 base pairs.
That's what I put.
Right?
Yeah. Do I put the BP as well?
No, I don't think so. Let me just double check.
Yeah, because that's what I put too, and I just put the number. I didn't put base pairs or anything, and I got it right. So
Oh, okay. I put
So,
401 BP.
yeah.
Well, I can just go back through that and adjust the grade accordingly. That's not a big deal. It's often in because Blackboard isn't particularly like sentient or anything, which is maybe a good thing. But in the, I'm sorry, I'm just kind of opening up that folder while I talk as well. Yeah, but so if it's just a number and you put letters in there too, it grades it wrong for some reason, even though you put the right information in there. Oh, I'm like, bloody hell. Did you put a space between 1 and the B?
No, I just put 401 BP.
That's really funny because I've got 3 different versions: 401, 401 space BP, and 401 space base pairs. And so you've found a 4th version. So I'll just add that 4th version. It's like every time I do this, and you're not wrong. It's just to be able to get Blackboard to grade it for me, which is nice. You have to put in kind of every permutation, and I'm actually doing this kind of as we speak. So hopefully this will automatically regrade yours and give you the points.
I could take the second attempt and just put 401.
No, I'm just kind of curious to see if, all right, go see if that gives you the right answer, because it will automatically regrade all the attempts.
Okay, let me check.
It's just a little easier than me going through them all manually. Though if you still have problems, I don't mind doing that too.
Yeah, now it's 50 out of 50. Thank you.
Oh, thank you. It's like, I can think of all the different permutations I might put, but that isn't necessarily all the ones that all of you might put. So it just makes it better, prevents these kinds of hiccups in the future. But yeah, if you ever have any problems like that, just let me know. And they're easy to fix.
Cool. Thank you, Professor.
No worries. Anyone else have any other questions? Queries? Pontifications? All right, so I'm going to get off and upload all the material from today so that you all have that to hand. And good luck in the exam, whenever you might take that, today or tomorrow. There aren't tricks in there, but you do have to think carefully about what I'm asking you, as always. All right, so have a great weekend, everybody. I shall see you next Tuesday. And then we're most definitely on the home stretch. Not much left to do. You too. Have a great weekend. Bye bye.